Hi, my name is Chacon, and in this video I will present my Capstone project for the Coursera's Advanced Data Science Specialization. This video has three sections. In the first section, I will talk about the use case and include a short demo of it. It is aimed to stakeholders and non-technical audience. The second section is an overview of the architecture used to host the web app. And finally, in the third section, I will go through the Jupyter notebook I used during the development of this project. The use case I choose is pneumonia detection and classification. With the current global crisis caused by COVID-19, medical centers are overwhelmed by the amount of patients. Therefore, a tool that allows technicians to perform pre-screening of the X-rays will ease the workload on radiology experts. The developed tool is a web app. It allows the technician to classify images by either uploading the sample or using a URL in case of the sample being stored in a remote location, for example, an FTP share. The application shows the probability that the sample shows pneumonia signs or not. If positive, it also shows the probability of being viral or bacterial. It also shows a score that indicates how good the sample is. This is useful as it will let the technician know if the provided sample might not have the correct orientation, is not centered, or simply the quality is not good enough to obtain good results. This is the diagram of how the application is hosted in IBM Cloud. It uses 6 different services. Jupyter Notebooks allow data scientists to perform experiments, test, and deploy models. Cloud Object Storage, it stores models and training data. Machine learning models, we use these to run models used for pneumonia detection, classification, and sample scoring. Cloud Foundry apps, this is where the web application is hosted. Deep learning experiments, to run scheduled retraining of the models with newer data. Cloud Functions, to trigger model retraining and assess their performance. A new model is only deployed if it performs better than the existing one. This is a working demo of the web app. It provides two options. Either you upload files from your computer or provide the URL of the sample you want to test. Let's try some samples without pneumonia. As you can see, with each sample, the detection is very confident, as the probability of pneumonia is very low. Additionally, the web app provides the score of the sample. In this case, these images are from the original dataset, so it is expected to have high scores. Now let's try some samples with pneumonia. Once again, the detection gives high probability of pneumonia. However, the classification between viral and bacterial pneumonia is not as precise as the detection. Let's find some bacterial pneumonia samples. Once again, the classification is not very confident. The reason for this behavior will be discussed in the technical part of this video. Finally, let's try some bad samples. Now we can see that the app still attempts to detect and classify the image. but now it warns the user that the quality of the sample is not good. Let's also try a sample from the original dataset that has been rotated 90 degrees. As we can see, the score is low. Interestingly, the prediction seems to be accurate. For this project, I used the chest X-ray pneumonia dataset from Kaggle. It contains over 5,000 images labeled as normal, viral, or bacterial pneumonia. In the link below, you can find more details about it. Now I will present the Jupyter notebook used to develop the web app. Since this is an image dataset, the first step to check its quality is by taking a look at some of the samples. As we can see, the first problem we have is that their dimensions are different. Also, there are some R letters randomly placed around the samples. We can fix that by adjusting the images to the desired size. In this case, our neural network expects images of 224 by 224 pixels. Since the samples are not squared, we cannot simply resize them as it will affect their shapes. Instead, we crop the sides for horizontal samples or top and bottom for vertical ones. Also, since the area of interest is mainly in the center of the image, I decided to crop all borders by 5%. This also fixes the problem of the R letters as it leaves them outside. The results are now equally sized, but now we can see another problem. The samples are not equally exposed. In other words, the samples either are too dark or too bright, and some of them lack of contrast. In some cases, so dark it is even difficult to see the vertebrae. To solve this problem, I used sklearn min-max scalar. Now we can see that all the samples are more even, with better brightness and contrast. Now we proceed to load the entire dataset, applying all the transformations mentioned before. After that, the first aspect we should check are the distribution of classes in our data. As we can see here, our dataset is imbalanced, so we will have to take this into consideration when we train our model. This is not a tabular dataset, so we don't have features to compare. Instead, we can use metrics like brightness to compare differences between classes. Here we can see that on average, normal samples are darker. This is expected as the author of the dataset explains that healthy lungs don't show any opacification. Another way to visualize characteristics of an image is by looking at its histogram. In this case, I calculated a 50 beans histogram per image. In this plot, I show the median per bean of the classes. We can see the difference between normal and pneumonia cases is more evident, while beans on the far right side of the plot mark the difference between types of pneumonia. By combining both the mean and histograms, we now have 51 different metrics, which seem to be good enough to train some linear models. I have to clarify that while some of the features seem to be statistically significant, it will be too naive to use them in a real-world project, as they pretty much only capture brightness. I created them simply for exploration and to have some tabular data to train traditional algorithms. Currently, the best way to deal with images are deep neural networks as they can learn features like textures, borders, and shapes. For this project, I compared three different algorithms. The first is linear regression implemented with Keras. The second is gradient boosted trees implemented with CatBoost library. And finally, a deep neural network, in this case, exception from Keras, and using weights trained with ImageNet. For the performance indicator, I choose ROC AUC because it allows to compare the overall performance between models. It is also especially helpful for imbalanced dataset like this, as a classifier that always classifies a positive will get high accuracy. As we can see, for the detection, the deep neural network performed exceptionally well, while the two linear models only achieved around 83. Additionally, I trained another two deep neural network models. The first is just another exception model trained only with pneumonia samples. It is used to classify pneumonia as viral or bacterial. The other model is an autoencoder trained with the entire dataset. It is formed by two models, the encoder, which will create an embedding that represents an image, and the decoder, which takes the embedding and attempts to reconstruct the image from it. This model will be used to measure the quality of the samples. Once trained, we can use it to calculate the mean of all vector representations in our dataset, which could be considered as an average representation of a desired sample. Then, we can encode new images and measure the Euclidean distance from the mean vector. We can also use the decoder to try to reconstruct the images from the embedding as shown in the right column. In this case, we are using new images, so the distances are big and the reconstructed images are not that good. In this case, these are the reconstructions from images from the dataset. As we can see, the distances are much smaller. In order to know how far an embedding should be to be considered a wrong sample, we can take a look at the distribution of distances for the entire dataset and set a limit based on how many standard deviations an embedding is away from the mean. Based on that, we can create a scoring function that returns a value between 0 and 100 based on the p-value. These are the scores of some images. As we can see, the more different the image is from a good sample, the lower the score it gets. That is all without going too deep into details. If you would like to take a look at the code, you can find the link in the description. Thank you for watching.